Welcome back, students. So in the first part, we have understood that by the help of the tryptophan and the aporepressor, when active repressor is made, that can bind on the operator region and can block the tryptophan operon, and that is why trypoperon is known as repressible operon. I hope you understood the reason why trypoperon or tryptophan operon is known as repressible operon. And for the repression, there are two pathways. Number one, by the help of operator repressor, like when repressor is bound on the operator region and can block the tryptophan operon, that is one method. And number two is by this leader region. Okay, so number one, through operator repressor, and number two, by the leader region. I hope you remember the leader region on the tryptophan operon. Before this, there was the promoter, there is the operator, and there is the leader region, and then the 5-tryptophan operon, and then the 5-tryptophan producing enzymes genes were present, and this was the leader region. Now we will come to this leader region very nicely. You can see in the leader sequence there are 4 regions, region 1, 2, 3 and 4, and these 4 sequences, they are complementary to each other. This leader sequence has 4 regions, region 1, 2, 3, 4. And once the leader sequence is transcribed, the mRNA, they can bind with each other. They can form alternative secondary structures by complementary base pairing. Okay, so from this leader region, mRNA is produced. And these mRNA, they can form this alternative secondary structure by complementary base pairing. Region 1 can pair with region 2, region 2 can pair with region 3, region 3 can pair with region 4. And when they bind in this fashion, they give a very important signal. When 1 pairs with 2, it gives a signal of pause. When 2 pairs with 3, it gives a signal of anti-termination. Anti-termination means it will not terminate, it will go on. And when 3 pairs with 4, it becomes a termination signal that will specify that lac operon will be turned off. Okay, and if you see this leader region, there is a start codon AUG, and in region 1 there are 2 TRP codon UGG, 2 TRP codon present, and after that 1 stop codon UGA, and there is then region 2, then region 3, then region 4, and then another start codon, that is AUG. So this is the overall structure of the leader region. So this is the leader region, and from the leader region the mRNAs are made, and the mRNAs can form alternative structures by complementary base pairing. And what they define, now we will come to this. You know that in case of bacteria, as transcription and translation are coupled. Coupled means translation will begin as soon as transcription is started. Like, for example, you know that in case of bacteria E. coli, there is absence of nuclear envelope. And for this, translation and transcription can go on simultaneously, that is together. So for example, suppose this is the loop, this is the transcription loop when DNA unbinds or unwinds and mRNA is started forming. So as soon as the mRNA is forming, ribosome will bind on the mRNA and will start producing proteins, you know. Okay, so that is why it is known as transcription and translation are coupled. As nuclear envelope is not present, so the RNA molecule need not to transport from the nucleus to the cytosol for translation. As soon as transcription is started, translation will also begin. So what happens in this case? The leader region is transcribed at first. So for example, this is the leader region, and the leader region, you know that I have told you and I have shown you also, the leader region is the first, then the consecutive TRP operon genes were present. So for example, leader region is transcribed first and result region will be transcribed first. When the leader region is transcribed, this transcription will give a signal to the rest of the RNA whether it will be produced or not. So listen carefully. So this is the DNA, and from the DNA the leader region is transcribed. The leader region will be transcribed at first because the leader region is present upstream of the 5 structural genes. So once the leader region is transcribed, ribosome will bind on this RNA to produce polypeptide from this leader region. Okay, and after that the rest of the 5 structural genes will be transcribed. Now here is the case. Here during translation, this translation will give the signal to the rest of the DNA whether the transcription is needed or not. So the first condition is when tryptophan is present in high amount. If tryptophan is present in high amount, when the ribosome is coming and started translating, you know, ribosome scans the reading frame and picks up the correct amino acyl tRNA and attaches with it. So if enough tryptophan is present, there will be enough tryptophan tRNA. You know, this tRNA will be conjugated with tryptophan and ribosome will bring it. Okay, if enough tryptophan is present, then ribosome can translate easily with tryptophan tRNA, and ribosome will continue the translation procedure, and it will be on region 2 after the stop codon. After the stop codon, the translation will be stopped and it will again bind on 2. Then region 3 and 4 will be free to pair with each other, and that will give the signal to the rest of the DNA that transcription needs to be turned off. Okay, that is why it is known as translation mediated transcription termination. It is also known as translation mediated transcription termination. Why? Because here the ribosomes, when they are translating, this translation is giving the signal to the DNA the whether the 5 structural genes need to be transcribed or not. So this will signal the DNA whether rest of the mRNA will be produced or not. Okay, that is why it is known as translation mediated transcription termination. Translation is helping to give the signal to the transcription that whether transcription will be terminated or not. So this will be the case in case of tryptophan abundance. When tryptophan is present in high amount, you know, when tryptophan is present in high amount, ribosome can translate these regions easily. So region 1 will be translated easily and it will go to region 2, and then region 3 and 4 are free to pair with each other, and this pairing will signal as termination. Okay, so the tryptophan operon will be terminated at this case. But what will happen when there is a starvation of tryptophan? What will happen when there is a shortage of tryptophan? So then the same case will happen. Then there will be less tryptophan tRNA. less tryptophan tRNA, then ribosome will halt on this codon. Ribosome will not get enough tryptophan tRNA for this procedure. Ribosome can't take enough tryptophan tRNA for this procedure for translation continuation. So ribosome will be halted on UGG. There will be no tryptophan tRNA present. So if ribosome is halted on region 1, what will happen? Region 2 and 3 will be free, and then region 2 and 3 will pair with each other, and what will happen? It will signal as anti-termination. Anti-termination means transcription will go on. Then this translation will signal the DNA that transcription needs to be continued. The transcription will continue because there is less amount of tryptophan present, more tryptophan is needed. So this translation is giving signal to the transcription system that tryptophan is needed, so continue producing the RNA. Okay, so that is the anti-termination signal. So these are the two cases. This procedure is also known as attenuation. Okay, attenuation, because this is happening because of the attenuator site. This leader region, this region is also known as attenuator site. So that is why this procedure is also known as attenuation. Okay, so I'm giving you quick revision. So I'm giving you a quick revision for the model of attenuation. In this case, whenever there is a tryptophan starved condition, that means tryptophan is present in low amount, what will happen? The ribosome will halt on region 1 because there is less tryptophan tRNA present. So tryptophan tRNA will not be present to equip with this ribosome. So ribosome will be halted on region 1. Then region 2 and 3 will free, and they will pair with each other, and that will signal that transcription anti-termination, the transcription will continue. Okay, but when there will be tryptophan high in amount, that is tryptophan non-starved, the tryptophan is present in high amount, what will happen? Ribosome will continue translating this region. Ribosome can continuously translate this region because there will be enough tryptophan tRNA. Enough tryptophan tRNA to equip with this ribosome. So tryptophan tRNA will be present in enough, so this UGG will be translated, and then ribosome will halt on region 2. If they halt on region 2, then region 3 and 4 are free, then region 3 and 4 will pair with each other, and that will signal to stop the transcription. Okay, so in nutshell, it depends on the level of tryptophan tRNA. Level of tryptophan tRNA decides whether tryptophan operon will be turned off or turned on. Because if tryptophan tRNA is present in high amount, that means tryptophan is present in high amount, then what will happen? Tryptophan can block this operon by two means. Number one, tryptophan can bind with the aporepressor and can block the operator. That is number one regulation. And number two regulation, tryptophan is high in amount, so tryptophan tRNA is present in high amount, so these codons will be translated easily and it will bind on region 2, and then the region 3 and 4 will be free. 3 and 4 will pair and will signal the transcription that it needs to be stopped. But if tryptophan is less in amount, there will be no tryptophan to bind with aporepressor to repress the operator, to bind to the operator. And as well, in case of this regulation also, there will be less tryptophan tRNA present to bind on UGG region to cooperate with ribosome. So ribosome will be halted on region 1, then region 2 and 3 will present backend, and they can then pair with each other and can signal the transcription anti-termination, that is, they can signal the transcription will be continued. So this is the overall regulation of the TRP operon. There are two means that whenever tryptophan is high in amount, they will bind with aporipressin and will block the operator, or what they can do, they are present in the tryptophan tRNA form, so the ribosome can translate it easily. Then region 3 and 4 will be free, 3 and 4 will pair with each other and can block the transcription. And another, when tryptophan is present in low amount, it can't bind with the aporipressor to block the tryptophan operon, or the ribosome will be halted on region 1 because the tryptophan codons can't be recognized by tryptophan tRNA, so the one will be blocked and 2 and 3 is free to pair with each other, and that will signal the transcription that yes, you continue transcription because tryptophan is present in less amount, more tryptophan is needed for the cell. I hope you have understood this tryptophan operon as well. If you have any queries, please let me know in the comment section, or you can mail me anytime. I wish you a happy learning. Fight for your dreams will fight for you, students. Thank you.